I thought I'd give a quick intro to what the Flatiron Institute is. We're right next to the Flatiron building. So this is the Flatiron building, that's our roof. And it's a lovely place to be. And it's a computational research institute funded by the Simons Foundation. And we have biology, mathematics, astrophysics, neuroscience, and quantum physics. And our group is joint between the Center for Computational Biology and the Center for Computational Mathematics. I myself am primarily in the Center for Computational Biology, and our group is called the Structural Molecular Biophysics, and it very much fits in well with the topics we're discussing here this week, and it's been a very exciting program so far. And as you might have noticed from Pilar's talk, what we're mostly interested in are molecular dynamics and methods development in cryo-EM, and also the interface between those two. And in my particular case, I'm coming at it less from a method side and more from a biology-motivated side. So actually, I'm very interested in trying to understand biological temperature sensing. I mean, we've already seen a few people today, and yesterday and Monday, talk about the TRPV1 dataset as a representative data set from cryo-EM because it played such a key role in the resolution revolution. I'm here to tell you a bit more about what that protein actually does and why it's really, really cool. But actually understanding it is a little bit difficult, and in the process I'm going to talk to you about a lot of the other things that people have already introduced this week, improving pose estimation, and in this case I'm going to talk specifically about using quaternions to do that. Talk a little bit about noise, and then finally end talking about heterogeneity and how that can really help us understand these biological temperature sensors. And here's a visual version of that outline, which I'll keep popping up as we go along to keep you situated. And yeah, as I said, I'm most interested in understanding biological temperature sensing from a molecular standpoint. So what is a biological temperature sensor? We understand physical thermometers. We have them in our cells, in our cell phones. We also have them in our cells. That's what I really want to talk about. In our cells, right, we have these temperature-sensitive resistors, and you can tell that. But actually, how we understand how biological thermometers work is not quite as straightforward. They're constructed of proteins, RNA, DNA, and there's a few of them that we understand, RNA thermometers, and various indirect sensors that detect temperature through like bilayer deformations or DNA bending. But here I'm going to focus mostly on intrinsic temperature sensing, where this property is intrinsic to the protein itself. And so the kind of poster boy for this kind of protein is the TRPV1, which is also a capsaicin-sensitive ion channel, in addition to being heat-sensitive. So it's up here and is activated at 42 degrees Celsius. It's an ion channel that opens at that temperature, allows cations to flow through, but it's also closely related to TRPM8, which is a cold-sensitive, menthol-sensitive channel. And you may know that last year, David Julius got the Nobel Prize for discovering these temperature sensors. And they're also activated by other things, such as spider toxins and pH. And understanding the molecular mechanisms of these proteins is still something we are trying to figure out. And the implications for this are potentially high, as potential pain therapeutics could be designed if we can figure this out. So you could actually go to your pharmacy right now and get capsaicin cream if you wanted to topically desensitize your pain neurons. Sometimes doctors prescribe this for patients with arthritis. But as of yet, there's no pill you can take that targets your TRP channels because it also affects your basal temperature regulation. And in clinical trials, people will get hypothermia when they try to design this kind of drugs. So understanding temperature sensing is not just cool and fundamental, but also has potential implications for real-world applications. So if we're thinking about studying these proteins via cryo-EM, how important is the cryo part of this, given that these are temperature sensors? And so there's some debate, I think, about these timescales, but this is what I've put on this slide. So for TRPV1, its estimated timescale of heat-dependent opening is around 2 to 5 milliseconds, while the estimated time for vitrification is less than a millisecond. So hopefully we'll be okay. But there's other questions here, such as like the column of air above the liquid ethane. Is that going to increase the amount of time that these samples are exposed to very high temperatures? And these are things we don't really 100% know yet, but there's been some promising work. For example, this Nature paper from 2010, where we could see in the ribosome shifting of the populations as a function of temperature before vitrification, which I see as a promising sign that we could study temperature sensors effectively in this way. But there are problems. So in the Sasha Sobolevsky lab, they have actually looked at TRPV1 at higher temperatures here, 37 degrees before vitrification. But they find that the majority of their particles represent a heterogeneous conformation. And this is consistent with other people who have tried to do this. It's just difficult to get real conformations out of these kinds of samples. And then lastly, another aspect of the cryo and cryo-EM is what's the ensemble that we have represented in our vitrified sample. What is actually happening during vitrification in this ice? And this is a nice computational paper here that starts to ask these questions and provide some answers. But I think there's a lot more that we could do to really understand these questions better. And so another aspect of biological temperature sensing is like that when we often think about temperature, we just think about it as flattening the landscape, similar to what I showed in that Fisher et al. Nature paper here. So where you can just, by increasing the temperature, get a better sampling of your conformational space. But in fact, and we don't know yet if this is the case for trip channels, but in fact you could easily have a temperature-dependent free energy, and which changes this picture to something more like this, where we have really a non-trivial relationship between our free energy landscapes between two different temperatures. And I think this is just nice to point out, because I think we don't always think about it in this way. I think people have talked about temperature just as like Brownian motion here, and that's not always the case. There are many more complicated things that temperature can do to our systems. And so despite the fact that despite the fact that we have actually lots of structures now of trip channels after the resolution revolution, we still don't understand their temperature sensing property. And one of the most compelling frameworks to think of this came from this PNAS paper in 2011, where actually if you think about a change in heat capacity between the open and closed states, you can start to explain both heat and cold sensing proteins. And here this means that we have a temperature-dependent enthalpy and entropy, because they're both dependent on heat capacity. And this relates also to experiments that have been seen where we have very high positive changes in enthalpy and entropy for heat sensing channels, and large negative changes for cold sensing channels. So we know some about the thermodynamics of these proteins as is. And this model also relates well to what we already know about protein folding and changes in heat capacity for protein unfolding. And somewhat as a subsequent follow-up to this paper, and the fact that there's a lot of experimental evidence that just has found a lot of individual point mutants that affect trip channel temperature sensitivity, and we've never found the temperature sensitive module in these channels. The general feeling in the field is that this is more a distributed property of these proteins, and this can fit well into the model of our heat capacity, and that we can have about 20 residues exposed to solvent per subunit in the trip channels, and that would roughly explain this kind of heat capacity difference and potentially explain their temperature dependence, and also a distributed model of temperature sensing. So that, I hope, convinces you that understanding the heterogeneity of these proteins will help us understand their function. And now I'm going to go deep into some nitty-gritty details about quaternion pose estimation, which we heard earlier can be intrinsically linked to actually estimating conformational heterogeneity from Tristan's and Fred's talks yesterday. So yes, quaternion pose estimation. So here I'm going to think of this in a very, very simplified framework. We're just looking at 3D point cloud matching or 3D point cloud to 2D projection matching, where we literally all we have are XYZ points in space. And this section of this talk actually has two subsections. The first, we're looking simply at 3D to 3D point cloud alignment. And here this is a kind of an ML motivated framework for this. And the second, we're actually looking at the more cryo-EM relevant question, where we have instead a 2D projection of our rotated 3D point cloud. Yeah? Yes, this is an important question. The first point I'm going to make here is that this whole framework is to explain to you our new adjugate quaternion framework, and the second thing I have right here anticipates your question, which is that in both cases we are considering the correspondences are known. So we know exactly, and here we have this, do I have a mouse? I do. We have these dotted lines to show we know we have ordered points, we know which point corresponds to which point. So similar to in a protein, we know alpha carbon 1 matches the other alpha carbon 1 in our two proteins that we want to align. So this simplifies the problem a lot and makes it mathematically tractable, but it's kind of a toy system for which we can start to understand limitations of estimating quaternion rotations in this problem. This might come up again, but feel free, but please do keep that in mind, it's important. Yeah. So why quaternions? One reason is that this was kind of a COVID project, and it was a collaboration with my dad, who literally wrote a textbook on visualizing quaternions. And when I started to talk to him about cryo-electron microscopy, he just was really enthusiastic about how we could use quaternions to better understand these questions we have about pose estimation in cryo-EM. But there are fundamental reasons why using quaternions is better. It's easy to produce uniform statistical distributions of our orientations and find smooth paths between orientations. And we have minimal discontinuities compared to Euler angles, which suffer from things like gimbal lock and have very, concrete discontinuities going from 2 pi back to 0, can mess up how you think about your space and your continuous rotation space. But I will say, while quaternions have minimal discontinuities, I actually am going to be talking about some discontinuities that we found in quaternions and how we can help get around problems with those. So our first part I mentioned is based on a machine learning quandary. So despite successes that we've had by people, for example, here at this conference, of machine learning for pose estimation in cryo-EM, CryoDragon 2, end-to-end orientation, cryo-AI. There's still questions about how we can better represent orientations and explore SO3, and there's still pathologies and local minima. And outside of cryo-EM, neural networks have shown to have some trouble predicting rotations in 3D space. And this whole project was basically motivated by finding these two papers, this 2019 paper from CVPR, which talks about the continuity of rotation representations. And in this case, their use case is trying to look at kinematics in robotics, and they found that using their 6D representation of orientations rather than quaternions performs better. And then this other Peretrukin paper, which is just on arXiv, shows another framework for looking at this problem. And so we found these papers, and we wanted to understand them a bit better because they had a kind of interesting result. But just to give a more concrete explanation of their setup, it's very simple. As I mentioned at the beginning, you have a reference 3D point cloud, and then you have a rotated version of that point cloud with added noise. Here the blue plane is that rotated version. And then what you want to do is you want to learn the quaternion that's the minimal rotation between those two 3D point clouds. And these are the results that these two papers showed. And in both cases, quaternion is in green. And in the Joe paper, their best result was this 6D in red, and then in Parachukin they have this A matrix in blue, which seems to improve even further on the Joe CVPR result. And in this case, the network is relatively simple. Takes on this point net structure from the CVPR 2017 paper, and in this case, X is 25 points in 3D space, and they train on 500 sets of data and test on 100 sets. But if we found out that their results, and here we've actually just taken the code from the Parachukin paper and run it ourselves, and we find that the results are an order of magnitude worse than if we just do RMSD, which is the analytical solution to this exact problem. And neither of these papers really mentions the RMSD solution to this problem, and we found that kind of startling. But you know, it remains to be asked, why is it that this neural network actually does not do well in this problem, and why is it actually that quaternions are doing the worst of all of these different frameworks? And so just to go in a bit more detail of what exactly this RMSD calculation is, other people know it as Cobb's or the generalized Procrustes problem. It's been like reinvented multiple times in aerospace and biochemistry. And I think the earliest solution to this problem takes our same setup as in the machine learning problem, and you just do SVD, and you can get exactly the optimal rotation matrix between these two sets of point clouds. And then in 1987, Horn also proposed a quaternion-based solution to this, where you can just use q.m.q, where m here is a matrix called the profile matrix. And here you can also solve the same problem by using the adjugate of this M profile matrix, which I'll go into in more detail in a second. So basically our two main questions here were, why are quaternions not performing well, and can we get down to RMSD accuracy with a neural net? And so to think more concretely about that first question, why are quaternions not doing well, we actually, racking our brains about this problem, found that if you just think about two representations of your rotation matrix, and here we're just looking at a 2D rotation matrix for simplicity. So here you have the quaternion version, and here you have that same rotation matrix as an angle representation. And actually in the next slide, I will use the simplified version of these same equations. And so if we set these two versions of the rotation matrix next to each other, equal to each other, we find that there's actually a discontinuity. And that at some point you actually end up dividing by 1 plus, or the square root of 1 plus cosine theta. And this means that you end up dividing by 0, where cosine theta is -1, or where cosine theta is 1, in this case down here. And this means that if you actually work in quaternion space to try and go from some xy set of points to your quaternions, you end up possibly going through this discontinuity. However, if you stay in an unnormalized state, where you look at these rings out here, the blue and the red, and you don't do this division by the 1 plus or 1 minus cosine terms, you don't ever end up finding these discontinuities. And so you can get full coverage of your space without discontinuities by staying in this unnormalized kind of expanded space of the quaternions. And here, just to show what is full coverage and what these discontinuities are in 2D space, and then we can also do the same thing in full SO3. It looks similar, but it's kind of harder to visualize what these discontinuities are. And so this is kind of where we started thinking about this unnormalized version of the quaternion. And it actually turns out that you can get this out exactly from the Horn way of looking at the RMSD, which is just an eigenvalue problem. And So if you look, I don't know, so far not a lot of the people I've talked to about this project with know what the adjugate matrix is, but it's a matrix composed of the transposed factors of the matrix. So your adjugate of a matrix is composed of that matrix's transposed cofactors, and it's defined by this equation here, and has the convenient property then when we think about it in terms of the characteristic equation, the adjugate matrix ends up being the eigenvectors for that problem. And so in this way we have then the adjugate, the quaternion adjugate ends up being just this unnormalized version of the quaternion. But it still allows us to solve this horn problem to match these two 3D point clouds together. And so in this way, we can get around these discontinuities in quaternion space. And here we just did a little test, putting it back into the code, trying to learn how to align these point clouds. And we do see that using our adjugate quaternion instead of the quaternion, we go down from the green line to this cyan line. But we actually still are not down to the actual RMSD solution, and we're still trying to figure out how to get down that far. And with that, I will move to the second section of this pose estimation part of the talk, and talk now about, yeah, go ahead.
Just as a question, what is your motivation for actually looking for a learning solution to this when you have an analytic one that is better?
So the question is just like, why is it not working? I think the idea is that, right, if we're doing something like trying to estimate the pose of an image with machine learning, shouldn't we be able to estimate the orientation that matches two 3D point clouds, which we know we can get analytically? And are there features of SO3 and our SO3 representation that maybe we can be doing better to learn in that orientation space? So it's just kind of a toy problem where we know that we have a solution that we should be able to get, and that we're using to kind of think about how to basically minimize orientations in SO3. Sure.
Practically, what is the difference between the two solutions, kind of numerically or whatever?
The neural net solution and the...
Yes, and the theoretical solution. So how fast one is calculated, how fast the other is calculated, what is...
So the neural net solution is way slower. I mean, so the analytical solution is like, 3 lines of code.
What about noise? So
this is with noise, yeah.
Even with noise, it gives much better results.
Yeah, so...
why do we use neural networks?
Yeah, I mean, I wouldn't suggest using a neural network for this problem, but yeah, I mean, it's kind of part of the reason that I haven't really progressed on this problem recently is basically because of this question. Like, it's not a thing that anybody really cares about solving with a neural net, but we're using it as kind of this question of like, why are neural nets bad at this when we know there's a way to be better? Yeah.
So how does Earth mover's distance kind of work in this context? Like if I use that to align point clouds.
So I think Earth mover's distance is when you don't have the matched points. In this case, we have matched points, and so RMSD is going to give you like the solution. Yeah. Yeah.
When there was this one of your slides, you mentioned there's sort of like 4 solutions. Or this is like 4 lines from the adjugate?
So this is the adjugate. So this is just multiplying the quaternion by each of its parts.
Okay, so is there a connection with the double cover issue? Is there like more than double cover or no double cover?
So
in the sense that like, is the adjugate to a 3x3 rotational matrix 1-to-1? Or is there some redundancy?
Yes, so one thing to say, right? So the adjugate is unnormalized, gives you like unnormalized set of quaternions. And what we actually do to find the quaternions is we find the element here with the max norm and then normalize that. So this will give us the exact same quaternion that is the right answer.
And it's just unique. It just gives us one quaternion.
Yes. And
So it also has just a double cover.
yes.
Okay.
Yeah. And yeah, the way this loss function is written here, it gets around the double coverage issue. But there are also things to look at there. Yes. So basically, this machine learning adventure inspired a second part of this story, and really was what got us on the track to thinking about this quaternion adjugate, this unnormalized 4x4 quaternion, and actually led us to find an analytical solution for the pose estimation problem. So here, instead of aligning two 3D point clouds, we have a 2D projection of a set of points, and we want to know what the rotation was of the template point cloud to generate that projection, similar to cryo-EM. And in this case, this problem is actually harder. We don't have a solution like RMSD because our rotation matrix in terms of quaternions is here, but when we have the projection matrix, and here we're assuming you're projecting on the z equals 0 plane, you have to remove the last line of that rotation matrix. And that gives you less information to minimize. We don't have the same solutions that we do to the RMSD problem. But now that we started thinking about things in terms of the adjugate quaternion, we now express this projection matrix instead of Q0 squared, Q1 squared, etc. We now have new variables which we've called adjugate variables, which are just those same variables but renamed as Q00, Q01, etc. And they have a set of rules that they have to obey. But the nice thing about this is that it actually means that our least squares optimization problem is now solvable in Mathematica, provided you provide it with the right constraints to, as I mentioned before, for all of these different quaternion adjugate variables. And so here, for the first time, we're able to present an analytical solution for the 3D to 2D pose estimation problem with this R tilde rotation matrix composed of these determinant values. And in for this particular matrix, it only works for the noiseless case, because this is only a true orthogonal rotation matrix when there's no noise. But we can actually use this Q.M.Q formalism from Horn and Bar-Itzhak to easily minimize this to a true rotation matrix. And I'll show you the results doing that later. And just to show what these D values are, we've decided to start calling this the determinant ratio matrix, or DRAM approach. And these D values are just composed of our original input coordinates from our X, Y, Z, and UV point clouds. Yes?
So you're estimating the pose actually under which the projection happens here?
Sorry?
You're estimating the pose under which the projection happens. So the rotation, and then you have an orthogonal projection. Yes?
Yeah. Yeah?
Is that a non-convex problem?
Sorry?
That's a non-convex optimization problem you're solving, right?
So once again, because we have matched points, it makes it a solvable problem. And that's why we can get this analytical solution. Otherwise, it's a much harder problem. I agree. But yeah, so sorry, go ahead.
Do you have a sense of like what mathematics is doing? Like, is it solving a convex optimization problem where it's doing some things numerically, or is it just have like a complicated system of equations to solve that are more like a linear program or something? Do you know sort of what's happening at that level of this nesting complexity of problems of linear and cone problems and blah, blah, blah?
Yeah, I don't know. We could talk about it later, about like what's happening under the hood. I know like it's. Is
it wicked fast, or is it?
Well, so the thing is that if you don't use the right set of constraints, it will give you an answer that's like 40 megabytes long. And so, which is not very nice. But if you play around with the constraints and how you actually set up this minimization, then you can get it to give you this simple solution here. But there is like, it's not, and it takes like an hour or two to run. Yeah.
You know this related problem of you are having several such projections and you do not know the 3D structure and not the pose. This is called, in computer vision is basically looked at, is called structure from motion. Basically people will say like we can know which point is which in different projections, so to say, but we do not know what the 3D structure is and we do not know what exactly the projection dimensions are. And there's solution to that as well, that have some analytical solutions.
Yeah, that could be nice. I'm not familiar with that problem, but it could be a nice extension of this if we can talk. Yeah, I don't know much about that, but it sounds interesting. Yes, so we propose the DRAM solution to the 3D to 2D pose estimation problem. And here's just some results where we have sigma equals 0.1. And here we compare our tilde, which is the solution I showed you, which isn't a true orthogonal matrix in the case of noise, in black. And you can see the least squares here is lower. But our RBI, which is the Bar-Itzhak corrected version, which is a true orthogonal rotation matrix, still performs better than the initial rotation matrix that we used to generate the rotated point cloud. And so here, this is a really compelling argument that we've really truly minimized this rotation and are able to produce results that have not been seen before. Yeah.
What was sigma is like if the points are perturbed or something?
Yeah, sigma is a noise. It's just Gaussian noise added on to the XYZ points before the rotation. Yeah.
So did you try like a semi-definite programming to solve for the rotation for the 3D to 2D problem? Because basically you just have like, if I understand correctly, like linear equations for the rotation, right? And you have the orthogonality constraint. So it seems to me like that.
Like argbin.
That semi-definite programming would work like for like
yeah. So
with the 3x3 matrix would work really, really fast.
Yeah. So there are other things which give us basically the same solution as this RBI, but this is like as fast as you can get basically. But I think we can talk more about other things to compare it to. I think here we're trying to emphasize the like analytical solution. I think,
it's almost as analytical as you can get. I mean, for like with such a small variable.
Yeah, sure. Cool. So that concludes this quaternion pose estimation part that I wanted to talk about. And here we have the quaternion adjugate, which improves learning in some neural network contexts. And we propose this new determinant ratio matrix. And some follow-ups that we're working on is seeing if the quaternion adjugate improves in other machine learning contexts, learning in SO3. So for example, implementing it in cryo-AI and see if that helps some of their learning there. And then, I'm hopeful that while this is very much a toy scenario, that maybe showing these solutions, I can help improve other methods of solving similar problems in this space. All right, so after that, I have a bit more levity with thinking about some noise, and then I'll move on to thinking about heterogeneity in cryo-EM. So one of the things that we've talked about a lot at this workshop is the problems we have around noise. It's tough to even confidently and reproducibly estimate your pose, for example. And there are other things that maybe we can think about more concretely, like how CryoSpark has implemented colored noise, and possibly going back to Poissonian distributions of noise. Of course, it's easier to implement Gaussian noise, but potentially, especially when it comes to thinking about heterogeneity and these small motions that potentially are important for biological temperature sensing, maybe it could be useful to go back at this. And just something to think about. So for example, in this project that Wai-Xing has been working on, he's been simulating signaling and generating fake cryo-EM data from this structure. And if we add in the waters that actually are always in his simulations, we see that just adding in the waters produces enough noise that we can no longer really see where this protein is. And here there's no noise actually here except the waters. Of course, this is an unreasonably small protein for cryo-EM, and if you do it with something like apoferritin, you can see the protein still, despite the fact that there's waters there, but you still see this noise. And of course, there's all sorts of reasons to not care about this, but I think it's a nice way that we can think about how maybe MD simulations can help us understand better models for noise or explicit atomistic representations of water, or even in the case of tomography, other proteins that we're trying to consider as noise, but may not actually be when we're actually generating images. And so go ahead.
So just to understand, when we go back to the
Sure.
small molecule that you had,
Sure.
This one is here. Basically, you don't see it anymore because it is moving so quickly within the water.
No, this is just a snapshot.
In the water, which are sort of randomly distributed.
This is just one snapshot of the protein. It's not because of the movement of the protein.
So then it's because of the water molecules that are surrounding it? Basically?
Yes. So here we have a box of water molecules on all sides of this protein.
And that just hides it completely?
Yeah.
Okay.
All right. And so especially in the case of, where we're thinking about what's really in a cell in tomography, we have proteins which are actually way denser, or not way, but denser than water, more around 1.3, 1.4 grams per mil compared to 1 gram per mil for water. And here there's actually some evidence that the smaller the protein, the denser it is. So I just wanted to put this kind of vignette in here to think about noise, and I'm happy to talk to anybody who wants to think more about noise in the context of these problems. Yes, okay, but now to the fun part. We could talk about heterogeneity in cryo-EM and how that impacts our understanding of these biological temperature sensors. So one of the great things about cryo-EM and the resolution revolution is that we no longer need crystals of our proteins in order to be able to get high resolution structures of them. But we're still kind of in this paradigm where we're still looking at one single structure of the proteins that we look at in single particle cryo-EM. Not always the case, but we lose a lot of particles on the way to this high resolution structure. And so a few people have already talked about manifold embedding here, and this is the method I'm going to talk about in more detail to try and get us toward free energy landscapes of these biological macromolecules and allow us to not discard all these other states that we see in our data sets. And so this method was first proposed by Abbas Ormazd and Joachim Frank for this ribosome data set here. And more recently, they've shown it to be useful for understanding the conformational change between the two states of ryanodin receptor going from a ligand-bound to ligand-unbound state. And here, as kind of a nice proof of concept, they show that if you map these distances between these two residues, they have a very different conformational, very different change in their distances than if you were just to interpolate between those two states. And here is just a nice movie of this conformational change that we can see from these data sets and this analysis. And so the method of manifold embedding was also already described a bit in the previous talk. Distance calculations, diffusion mapping, then there's this NLSA step. But one aspect that was not discussed so much is that actually manifold embedding is performed on each projection direction at a time. So one of the first things that happens in manifold embedding is that the projection directions are clustered into sets that have approximately, where the conformational change is going to be the dominant signal compared to the orientation change. And so one of the key steps in manifold embedding is actually connecting up all of the manifold embedding results for each projection direction. And Suvrajit Maji produced a tool that allows you to do this automatically using an optical flow method. In practice in the ribosome and ryanodin receptor papers, this was all done manually, and actually in the work I'm about to show you, connecting up these projections was also done manually in this case. But it's nice to see this work from Suvrajit to see how you could do this automatically, and now the existing Python GUI allows you to do this with Suvrajit's toolkit, which is very nice. And so just to show this in a bit more detail, in his method, you pick anchor nodes where you define the conformational change to be in the positive direction, and then in other anchor nodes, for example, going like up to down in the case of these wings of ryanodin receptor, and you make sure that that is the same order of conformational change in all of the other anchor nodes that you describe over the orientation sphere. Yes. And so in practice, how that works is that there's actually these optical flow that looks at these projection direction movies and defines the direction of these conformational changes and links up all of these projection directions across SO3. Okay, and so I applied manifold embedding to the original Apo TRPV1 data set. And we were able to build kind of a pseudo free energy landscape and see motions, but not build volumes. The resolution for across this landscape was too low to do this. And so I was interested in doing this better, to do this for a newer data set with more particles. And so I ran this then on the TRPV1 bound with two different toxins and in nanodiscs, but then it turns out that actually the heterogeneity of the nanodiscs masks out any conformational heterogeneity of the protein that we can see there. And so there's other things we can do to try and mask out the nanodiscs, et cetera, et cetera. But it gets a little annoying because also the nanodiscs themselves are heterogeneous, and so you kind of ideally would want a heterogeneous mask on the nanodiscs. Anyway, this is still ongoing. And so we've also tried analyzing the same data sets with CryoDragon. It has a similar issue with nanodiscs dominating the heterogeneity. And then furthermore, when we're interested in free energy landscapes from these data sets, it's tricky to kind of get this from CryoDragon because of the non-identifiability of the VIE latent space means we really should not interpret these spaces as free energy landscapes. That can be said about other types of dimensionality reduction as well, but that's a larger question. And so furthermore, Evan Zeitz, who recently graduated from Joachim Frank's group, has proposed optimization to the manifold embedding method based on a simulated data set, which Pilar also talked about, and you can find a description of in the bioarchive of this HSP90 data set. And basically, the use of simulated data sets here was crucial to trying to improve manifold embedding so that they knew they were getting the ground truth free energy landscape back in the results from manifold embedding. And so here I'm going to kind of echo what Pilar was saying, is that it'd be great if we can have an apoferritin for heterogeneity, and I think both in the sense of simulated data sets and experimental data sets. So generating even better simulated data sets where we have real heterogeneity, for example, from MD. So here, for example, DeepMind has their own VAE, but instead of learning volumes, they're learning structures, and they actually used a folding at home data set of kinases, which I also analyzed and was involved in during my postdoc. And furthermore, we don't just need better simulated data sets, but really better experimental data sets would be amazing if we can really agree on something where we have potentially even validations from an experimental method such as FRET or NMR, whatever one could get from this. Yeah. And so to summarize this last part on heterogeneity in cryo-EM, there's still lots of challenges relevant to small molecules, small for cryo-EM, where the conformational changes we're looking for. So for example, this process of matching up all of the conformational changes over orientation space is especially difficult in the case of trip channels, where like it requires me to by eye actually see the conformational change that I'm looking for, which is not as trivial as it is in the ryanodine receptor case. And furthermore, validating these methods is still an open question that we're very interested to see what the community has to say. And this isn't just relevant to getting free energy landscapes, but also to quantifying errors in discrete heterogeneity as well. For example, as Sandeep Kalandolkar was doing in this time-dependent cryo-EM, where he had to run multiple runs of RelyOn in order to get these populations of different states of the ribosome. And because RelyOn is stochastic in the number of states it assigns to different populations, it's sort of not trivial to really do this, even in the case of discrete heterogeneity. And so I think any advances we can make in understanding free energy landscapes will also have an impact on general 3D classification and what are the uncertainties there when we try and get these populations. And so that's my last section of the talk. And so this is some take-home messages and questions for you all. There's still lots of problems to solve related to studying biological temperature sensing with cryo-EM. There may be some potential improvements here in estimating orientations in SO3. And I think there's some cool things we can do with simulated data, both from MD and thinking more about explicitly including things in our noise. And then finally, how can we validate our methods for obtaining populations from cryo-EM? So thank you guys very much for your attention, and especially thank you to the Flatiron and Malcolm Frank, who's been very supportive with this work, and Professor Emeritus Andrew J. Hanson, who contributed greatly to the quaternion portion of this work. So thank you for your attention.